Dear students, today I'm going to deliberate on the topic Trichoderma, a biocontrol agent. Pathogen, pests, and weeds are the enemies of agriculture and forestry. But this can be controlled by various methods. I will mention only two methods over here. By chemical method, by using chemicals like fungicides, pesticides, nematicides, and weedicides. Biological control, here one organism can be used to control the growth of another organism by exploiting parasitic or the predatory relationship. And this can also be done by using microorganisms like bacteria, fungi, and viruses, and also by using the botanicals. Let us look into biological control. Biological control is one of the methods of plant disease management. It forms an important component of integrated pest management. Now, what is actually biological control? This is another way of describing biological control of plant pathogen. It refers to the total or partial destruction of the pathogens by other antagonistic microorganisms. For example, Trichoderma, Penicillium, Bacillus, and Pseudomonas. You can see the pics over here. This is of Trichoderma, Penicillium, Bacillus, and Pseudomonas. So such microorganisms that are used to control the diseases caused by pathogens and pests like insects and nematodes are called as microbial biopesticides. Now, why do we need biological control agents? Let us compare with the chemicals, then it will be quite clear. Chemical pesticides, these are implicated in environmental and human health problems. It means that when the farmer uses this chemical pesticides on his farm, then it causes environmental pollution and it also causes human health problems. It is also very expensive, and these chemical pesticides have to be applied every time the farmer has to have his crop. Then also these chemical pesticides, they are toxic to both the beneficial as well as the pathogenic species. Now let us look into biological control agents. Now these are not at all toxic to human beings, nor are they toxic to animals. Also, they do not pollute the environment. Also, they are host specific and they are not as costly as chemical pesticides. That is, it is quite economical. Now let us look into one particular biocontrol agent, that is Trichoderma. A little bit about the history of Trichoderma. In 1671, Trichoderma was first studied in Germany. In 1794, it was identified by Persoon. In 1927, Gilman and Abbott identified four species of Trichoderma. In 1932, Winling, he was the first person to demonstrate that Trichoderma mm, can parasitize that fungi which causes the wilt of pigeon pea. So pigeon pea is our red gram. Now let us look into the taxonomical position of Trichoderma. Dear students, you know that when the fungal life cycle is being studied, there were some fungi wherein only the asexual phase was discovered. And it was not sure whether the sexual phase was present or not. So such fungi, they have been included in Deuteromycetes. Same is the story with Trichoderma. A sexual stage was studied at one time and later on the sexual stage was discovered. So it has got two ways of classification and also it has got two names. Let us look into that. So uh, the asexual stage has been classified as it belongs to kingdom fungi, division Ascomycota, subdivision Deuteromycotina, class Hypomycetes, order Moniliales, family Moniliaceae, genus Trichoderma. And when the sexual stage was discovered, then it was classified as kingdom fungi. Division is Ascomycota. Subdivision is Ascomycotina. Class Pyrenomycetes. Order Spirales. Family Hypocriaceae. Genus Hypocrea. So Trichoderma and Hypocrea are the name of one single fungi. Now let us look into the microscopic features of Trichoderma. So in this pic you can see that Trichoderma, the hyphae is hyaline and it is septate. How does it reproduce? So it reproduces by producing the conidia. So you can see the conidia, they are oval and they are uninucleate. Now these conidia are produced on the conidiophores. And these conidiophores, they are hyaline and they are branch. Trichoderma also produces thick-walled and resistant spores which are called as the chlamydospores. It also has got the phyalides which are hyaline, and you can see that they are flask-shaped and they have got an inflated or swollen base. In the center you can see a petri dish wherein there is a culture of the trichoderma. In the center you can see it is green in color. That is because it is showing the conidia, and these conidia are green in color. A few points about trichoderma. So trichoderma is a very effective biological means for plant disease management, especially of the soil-borne diseases. Trichoderma is a free-living and it is saprophytic fungus, and it is common in the soil. That is, it is soil-inhabiting fungi, and especially in the soil it is found in the root ecosystem, that is the immediate zone surrounding the roots, that is called as rhizosphere. Now you can see a pic of the petri dish with the trichoderma culture, and you can see a test tube slant with the trichoderma. Now the third point is that trichoderma is highly interactive, in which environments, in the roots, in the soil, and also in the foliar environments. That is the zone surrounding the leaves, which is called as a phyllosphere. There also it is very interactive. This trichoderma, it reduces the growth, the survival, and the infections which are caused by pathogens by different mechanisms. Let us look into the different mechanisms. They are competition, antibiosis, mycoparasitism, hyphal interactions, and enzyme secretion. So let us look into one by one. So this particular pic, okay, it gives you a glimpse of all the different mechanism of actions shown by trichoderma. So here's the trichoderma, which is asserting the biological control mechanism, and these are the various ways. That is, it shows mycoparasitism, it shows production of antibiotics, that is antibiosis. It is active in the rhizosphere, it competes for nutrients and space, it produces the different enzymes like chitinases and glucanases, it induces resistance in the plant, it is an endophyte, so it shows endophytism. It uh, makes the plant to tolerate against the stress, and it also helps in the root development of the plants. So this is a glimpse of all the mechanisms of action, so we can look into it one by one. The first mechanism of action is competition. So Trichoderma, it competes with the pathogen for the food and the essential elements. So it displaces and suppresses the growth of the pathogen population in the rhizosphere, and thus it reduces the disease development. For example, when soil treatment with Trichoderma harzianum spores was done, it was found that it suppressed the pathogen Fusarium oxysporum species. Now, the second mechanism is mycoparasitism and lysis. Now, Trichoderma is a fungi. It in turn it grows and it overpowers the growth of other fungi. So this phenomenon is called as mycoparasitism. Okay, and how it does it? It does by means of lysis. So here's a pic wherein you can see this is the pythium, the blue color. This is the hyphae of the pythium. And that which is coiling around this pythium is our trichoderma. So you can see how closely the coils are formed, and it has come into close contact with the pythium. And what happens is that wherever it comes in contact, the trichoderma comes in contact with the pythium, then it spreads out to produce the special structures which are called as the apressoria. I can show you a little bit. So you can see over here in the soil, the trichoderma is growing over here, and there's a pathogen. As soon as the pathogen begins to grow, this trichodermal hyphae approaches the pathogen itself and it forms the coil, and you can see how it spreads out. This is called as the apressoria. From this aprosuria, again multiple hyphae are produced. So this hyphae will penetrate the pathogen. Um, Other method of penetrating is by means of hostoria. And you know that the fungal cell wall, it is made up of the chitin and the pectin. So what it will do is the trichoderma, it also produces the enzymes, okay, special enzymes that is the lytic enzymes by name chitinase, glucanase, and pectinase. So this what they will do is they will actually degrade the wall, cell wall of the pathogen. Okay, and finally, it brings about it digests the cell wall of the pathogen and brings about the lysis. Okay, so this we will look into in a form of the point. So the antagonist Trichoderma hyphae, it either grows along with the host hyphae or it coils around it, and it secretes different lytic enzymes such as chitinase, glucanase, pectinase, and these are all involved in the process of mycoparasitism. Now, if you break down that interaction into different, into a number of steps, you can see the trichoderma grows towards the pathogen, that is by the process of chemotaxis. Okay, then it coiling of the hyphae around the pathogen takes place. Extracellular enzymes, that is chitinase, glucanase, and pectinase, are produced. Apressoria-like structures are produced, and with the help of which it penetrates the pathogen. Penetration happens even by hostoria. Then vacuolization of the pathogen takes place, and finally lysis of the pathogen takes place. Yes, students, trichoderma is also called as a hyperparasite. Example of such interactions are, Trichoderma hertzianum, Trichoderma viridae. They act against Fusarium oxysporum, they act against Fusarium roseum, Fusarium solanae, Phytophthora colocasia, then Pythium, Sclerotium rolfsi, and Rhizoctonia. So what it does it parasitizes the mycelia and it inhibits the growth and the spread of the pathogens. So here I've shown a pic wherein this hyphae belongs to Rhizoctonia solanae, and you can see how Trichoderma has completely coiled on Rhizoctonia, and it is actually strangulating it. This pic I've already explained to you. This is another pic. You can see the Trichoderma and coiling, and you can see over here the cell wall of the pathogen has already been digested. The third mechanism is by means of antibiosis. That is, it produces antibiotics. Okay, so trichoderma produces different metabolites. Some of them are diffusible inhibitors, like antibiotics and toxins are produced. For example, trichothiocin, sesquiterpine, trichodermin, and viridine. Along with this, even volatile inhibitors like sesquiterpene, alcohols, and ketones are produced. Okay, and these are excreted, and these have a harmful effect on the other organisms that act as the pathogens. So this is a pic about the uh, growth of this one trichoderma, like the antagonism. It's about the antagonism relationship that it shares with the pathogens. So you can see in this petri dish, you can see the wells are over here, and whatever white thing you can observe, this is the trichoderma. So in this well, these three wells, trichoderma has been inoculated, and the white what you see is the mycelium of the trichoderma. Now, after this petri dish is kept for incubation, you can see over here in this three wells over here, the pathogen has also been inoculated. After incubation, what do you observe over here? There is no pathogen at all. What has happened is the trichoderma has completely overpowered. It has not. It has prevented the growth of the pathogen itself. And in all these three wells, there is profuse growth of trichoderma. Now let us look into the next point, that is benefits of trichoderma. What are the benefits of trichoderma? So the first benefit is that it acts as a plant growth promoter. Now trichoderma The different strains of trichoderma, they solubilize the phosphates and the micronutrients which are present in the soil, and it makes it available to the plant. So the plant can easily uptake these phosphates and micronutrients, and therefore it can show robust growth. Their application to the plants, it increases the number of the deep roots, and thereby what happens is it increases the plant's ability to resist the drought. The more the number of deep roots, the plant will be able to absorb water more efficiently, and also it will be able to uptake the micronutrients and the various phosphates etc. more efficiently. So here you can see a pic over here. So this is the pic showing the root of a plant where trichoderma has not been used. So there's a very sparse growth of the roots. Whereas over here you can see the profuse growth of the roots because trichoderma has been used. Now this is another pic. So here you can see that these are the bean plants. You can see the roots of the bean plants where trichoderma has not been used. They're very, very poorly developed. Whereas when trichoderma is used, you can see a very robust growth of the roots. Now this is another pic to demonstrate the benefit of trichoderma treatment. So here's a plant, it is showing some diseased symptoms, and just look into the roots. The roots are very, very few. That is it has got sparse roots. Now trichoderma is growing wherever the root is there, and then in the rhizosphere region the trichoderma is growing. When some uh, hy- when some pathogen approaches the root, to attack the root of the plant, this this trichodermal hyphae, it by means of chemotaxis, it goes and it coils around the hyphae of the pathogen, and this process we've already discussed in the previous slides. So that is how it protects the plant from this pathogen. Similarly, it also makes available the water, also makes available the nutrients and the macronutrients. So after trichoderma treatment, what happens is the plant recovers and it shows very healthy growth, and you can see how much that is a very profuse development of the roots. The second point or second benefit is that about disease control. Trichoderma is a potent biocontrol agent and it is used extensively for soil-borne diseases. It has been used successfully against pathogenic fungi which are belonging to various genera like Fusarium, Phytophthora and Sclerotia. I, give, I want to show you some pics about few fungi which cause diseases to the plants. So this is Sclerotium Rousai, and it causes stem rot. So you can see the symptoms over here. And this is, you can see this is Fusarium, and this is, the tomato plants which have been attacked by Fusarium, causing the Fusarium wilt disease in tomato. Students, you can see over here that this is Colocasia, which has been attacked by the fungi Phytophthora. This is Phytophthora, and just see the symptoms when it attacks the plant. You can see these are the necrotic lesions which are there on the leaves. So if trichoderma is used, then it can control the attack by Phytophthora colocaceae. Now this is a very interesting pic. These are the plants of tomato. Okay, so tomato is attacked by different pathogens. See, Rhizoctona solanae, Pythium species, Phytophthora, the Sclerotium rolsi, Macrophomina phaseolina, Alternaria solanae, and Botrytis cinerea. These are the fungal pathogens, which will attack the tomato plant. But if you apply trichoderma to the soil, then it can control all these pathogens. Well, you can see the pythium. This is the pythium, And you can see Pythium along with Phytophthora infestans. These are the pathogens. And you can see over here, these are the normal or healthy seedlings. Whereas when it is attacked by Pythium, you can see the damping of disease takes place. Damping off of seedlings takes place. Then this is another pic showing the Phytophthora. This is the Phytophthora and it causes the disease in the potato plants. The third benefit is that trichoderma, they are biochemical elicitors of disease. What do you mean by this? See, trichoderma strains, they are known to induce resistance in plants. Okay, it produces three classes of compounds, and these compounds, they make the trichoderma uh, by which the trichoderma can induce resistance to the resist it sorry it induces resistance in the plants. So these compounds, they induce ethylene production. It brings about hypersensitive responses and also other defense related reactions in the plant cultivars. Also, the fourth benefit is, as we discussed, that trichoderma, it produces the enzymes called as chitinase enzyme. So what has been done is that the gene which is responsible for production of chitinase enzyme has been identified, and it is called as endochitinase gene. So this endochitinase gene has been isolated from trichoderma, and this has been then by means of genetic engineering, it has been introduced into tobacco and potato plants. So what has happened is now that the tobacco and potato plants, they are showing increased resistance to fungal attack. And these plants, in which the endochitinase gene has been introduced, they are called as transgenic plants. So these transgenic lines, they are highly tolerant even to the foliar pathogens like Alternaria alternata, Alternaria solanae, and Botrytis cinerea, as well as to the soil-borne pathogens like Rhizoctonia species. The fifth benefit is that it brings about bioremediation. Trichoderma strains, they play an important role in bioremediation of the soil, which has been contaminated with pesticides and herbicides. This means that, but due to continuous use of herbicides and pesticides by the farmers, what happens is the soil gets contaminated with this. So when trichoderma treatment is given to the soil, that is, it is added to the soil, this trichoderma has got the ability to degrade these wide range of insecticides, that is organochlorines, it can degrade organophosphates, and it can also degrade carbonates, and it can make the soil healthy. So this process is called as bioremediation. Sixth benefit is that trichoderma, it enhances, that is, it increases the yield of the crop and also the quality of the crop. Seventh is that trichoderma, it boosts the rate of germination of the crop plants. Then eighth point is that it causes an increase in the root length and the shoot length of the crop plants. Then tenth is that trichoderma promotes healthy growth in the early stages of the crop, and this causes an increase in the uh, dry matter, which is uh, uh, which symbolizes the healthy growth of the crop. The 11th point is that it provides natural and long-term immunity to the crops and to the soil. And the 12th point, which is most important, is that it augments nitrogen fixing. That is, in the soil, there are many organisms which fix the atmospheric nitrogen and make it available to the plants. So if trichoderma is added to the soil, then it will cause an increase in the ability of those microorganisms to fix the nitrogen. Now this is a pic wherein all the different mechanisms of action has been depicted. So You can see the growth of the plant over here before using trichoderma, after using trichoderma. Okay, so trichoderma competes with the pathogen. This is a pic before the treatment of the plant with trichoderma, afterwards. That is, here it is showing antibiosis, here it is showing mycoparasitism, and here that is, when a soil is treated with the trichoderma, it induces resistance to the plants, in the plants. It induces resistance in the plants, and it makes the plant itself to resist the attack of the other fungi. Trichoderma also acts as an endophyte, okay, and that is also beneficial to the plant. Now, let us see what are the different uses of trichoderma, where all, in which all diseases trichoderma has been used. There's a quite a big list. You can remember a few of them. So trichoderma can be used as a biocontrol agent in case of damping off caused by Pythium species and by Phytophthora species. Then it can be used in case of root rot caused by Pellicularis filamentosa. It can be used when there is seedling blight caused by Pythium, collar rot caused by Pedicularis rolfsi, dry rot caused by Macrophomina facioli, charcoal rot caused by Macrophomina facioli, loose smut caused by Ustilago sigitum, kernel bunch diseases, black scurf caused by Rhizoctona solani, then foot rots caused by this foot rots which are caused by the fungi in case of pepper and beetle vine, and capsule rot of several crops. It is also effective against silver leaf on plum, peach and nectarine. And in case of Dutch elm disease on elm's honey fungus, that is Armillaria amelia, it is also effective in botrytis, that is caused by botrytis cinerea. Trichoderma, polysporum, it also controls the decay of the wood. So you can see that Trichoderma species, it can be used to control several plant pathogenic fungi. Now let us look into the trichoderma formulations. That is, in what form or under what trade name or brand name trichoderma is being sold in the market. So important commercial formulations are available in the name of Sanjeevani, Guard, Niprot, Bioderma, etc. So these formulations, they have 3 into 10 raised to 6 colony forming units per gram of the carrier material. So this is a table which this is a table which shows the available biopesticides in the market. So Trichoderma virens, the first column shows the Trichoderma species, the second column shows the trade name, the third column shows the pathogens, the target pathogens, and the fourth column shows the crops on which it can be used. So Trichoderma virens, Trichoderma hertzianum, Trichoderma it is available under the trade names of Soil Guard, Plant Shield, T22, T22, okay, and the target pathogens are Pythium, Rhizoctonia, then Fusarium, Pythium, Rhizoctonia, and it can be used on ornamental crops, food crops, okay, whether they're grown outside or in the greenhouses. It can be used in case of cucurbit vegetables, flowers, and even in the nursery beds. It can be used for the fodder crops, for the bulb crops. Then it can be used in case of horticultural crops, okay, then in spices and legumes also. Mm, yeah. In continuation of this, it is available in the, under the trade name EcoFit, Supervisit, Soil Guard, Root Pro, Tuzal, okay, Agroderma, BioCure, Bioderma, EcoFit, Rakshak, Trichosan. Now, what are the Trichoderma species? Trichoderma viridae, Hertzianum virens, and here are two species together being used in Tuzal. So these are the various manufacturing companies. Now let us look into the methods of application. The trichoderma has to be used, but how has it to be used? What are the different methods of application? So one is seed treatment. So mix 6 to 10 grams of trichoderma powder per kg of the seed just before sowing. Then it can be used for nursery treatment. So apply 10 to 25 grams of trichoderma powder per 100 meter square of the nursery beds. Okay, and along with this, if neem cake and the farmyard manure is applied before the trichoderma is applied, then that will increase the efficacy. Then the trichoderma can be used for cutting and seedling root dip. So 10 grams of trichoderma powder is mixed with 100 grams of well rotted FYM per liter of water. And the cuttings, and the plants which are propagated by means of cuttings and even the seedlings, they can be dipped into this for 10 minutes just before planting. Then the fourth method is soil treatment. So 5 kg of trichoderma powder per hectare can be used, and this can be applied to the soil, and that is called as green manuring. Or what can be done is 1 kg of trichoderma can be added to about 100 kg of farmyard manure, and then it can be covered for 7 days with the polythene cover, and then it can be sprinkled with water every now and then, and this mixture can then be used in the field. That is how the soil treatment can be done. Or directly, the plant treatment can also be done. So, what can be done is the soil that is near the stem of the crop plant, that soil, it has to be drenched with trichoderma, 10 grams of trichoderma mixed with 1 liter of water. In that ratio, the drenching can be done. Now, finally, what are the advantages of using trichoderma, and what are the disadvantages of this biocontrol agent? So, advantages: trichoderma decreases the disease intensity. It reduces the use of chemical fungicides. It reduces the undesirable effects from the chemical fungicides. It plays a key role in integrated management of diseases. It is safe for the users and the farming community. It provides natural long-term immunity to the crops as well as to the soil. What are the disadvantages of biocontrol agent? There are quite few. Uh, They may have deleterious effects on non-target microorganisms. Sometimes what happens, the pathogens may develop resistance to trichoderma. And sometimes what happens is control of the target pathogen. Trichoderma will go and control the pathogen, but then what may happen is that some other pathogen may come and replace this. So it may lead to pathogen replacement. Sometimes what happens is due to seasonal and weather conditions may make the biocontrol agent effective. Yes, students, I have added a few more slides just for your reference. So you can go through these slides for extra reference. So this is also this slide also to show the mechanism of the action of trichoderma. This is also to show the mechanism of action of trichoderma. Well, thank you. That's it for now. Thank you, dear students.